Hi there. Welcome to the From Lab to Launch podcast by Qualio, where we share inspiring stories from the people on the front lines of life sciences. Tune in and leave inspired to bring your life-saving products to the world.
Hello and welcome to From Lab to Launch by Qualio. Glad you're tuning in today. I'm Kelly, your host, and I'm really excited about today's guest. Before we jump in, just a reminder to please rate the show and share it with any of your science nerd friends. We know you have some. Also, check out the show notes if you have a story or a product you'd like to share with us. Today, we're excited to have Dr. Jain, CEO of Sapient. Dr. Jain, or Mo, as he prefers to be called, is a physician scientist with nearly 20 years of experience in physiology, biomedicine, engineering, computational biology, and mass spectrometry-based metabolomics. He started and directed the Jain Laboratory at University of California, San Diego for a number of years, and that's actually where Sapient got started as well. You can read the full bio about Mo in the show notes. Sapient is one of the largest capacity biomarker discovery labs in the world and well on its way to transform biomedicine forever. Sapient has multidisciplinary team, sponsors, and therapies with biomarker guided insights. For all of our science and bio nerds listening in, I'm sure you'll be fascinated by the insights from Mo today. Thank you so much for joining us today, Mo. Welcome to the show.
Thank you so much, Kelly. My pleasure to be here, and thank you for having us.
To get started here, you have such an accomplished background, as our listeners can see in the show notes, but tell us briefly what interested you to pursue a career in pharmacology and bioactive metabolites in human disease.
Absolutely. I think my background is a reflection of more severe ADD than anything else. In that I'm searching for what do I want to do when I grow up. And I've had the distinct pleasure of being able to work in several different areas, both as a practicing physician, as a professor and a researcher, and now in industry with Sapient. This career really got started with a fundamental question of wanting to understand why some people stay healthy over the course of their life and why other people develop diseases and being best to help those people. And this is why I became a practicing cardiologist in my early life. And as that process evolved, it became really clear to me that even for all that we know about human disease, we really understand a very small portion of a very complex system, which is the human body. And we're really bad at predicting who's going to develop what disease over time. And we're even worse at predicting who's going to actually respond to a given drug. And the numbers are actually astounding as you go through the actual data, whether it be for clinical trials or real world evidence. Essentially, the best drugs work in about 50% of people, which is, again, a number that's just absolutely appalling to me. The fact that we're not able to diagnose disease decades in advance, when we know these processes take years or decades to actually come to fruition, is also just really not acceptable as a civilization, as a way of practicing. And so it was that desire to want to be able to evolve how we diagnose disease, how we develop drugs, how we distribute and whom we use particular drugs that gave rise to initially my research program and then ultimately Sapient.
Yeah, as someone who operates an industry from a quality perspective, of course, we're always looking at those kind of percentages. And I think the greater public really doesn't understand how much your individual biology plays into all of these things. Doctors prescribe us a drug, we expect it to just work. We don't understand why it doesn't. We get frustrated. We blame the company, whatever. It's crazy to me that people, maybe, I don't know, maybe we just don't do a good enough job in high school biology.
Yeah, part of it is an education issue, but part of it is also just how we go about deploying drugs.
Yeah.
The way we think about this is that everyone who has a disease is diagnosed as a universal grouping of individuals that, as you said, will all receive the same therapeutic for the most part. And we know there's huge variations within any disease population, both in how the individual developed that disease and whether or not they're going to respond to a specific therapy. Part of that has to do with the fact that, Kelly, you're a little bit different as a human being than I am. Part of it has to do that even if we have the same disease, our diseases are quite different from one another. So
Right.
being able to understand how we deeply phenotype disease, particularly at its earliest stages, and then deploy drugs in a way that are targeted to help us target our specific therapies is really the crux of this problem. And this is the goal of personalization of medicine in general. And it's always been a sort of a wonderful idea and a wonderful concept. And in certain therapeutic areas, particularly in the oncology space, this has really been transformative over the last decade. But medicine as a whole has not very much evolved in thousands of years, arguably. We still diagnose disease based upon a certain pathology. And it's a one disease, one drug type of relationship. And that's proven to not be correct.
Definitely. And it's fascinating to me too, right? My spouse has a particular fascination with all things microbiome and how that plays into all of this too. But I think as humans, we want to have that. Here's the single answer. And the whole idea of correlation isn't causation. There's actually a whole lot of factors at play. How do you keep from the gene becoming just the magic bullet answer as well?
Yeah, it's a good question. It depends upon what the underlying objectives are. And I'll explain what I mean by that, Kelly. Sometimes simple correlation can provide a lot of diagnostic information. And let me use an example of the good cholesterol, HDL. There's an abundance of evidence that shows HDL is not actually causal for protection from heart disease, and it's reading out other factors. But it's still an exceptional diagnostic for telling us who's at risk for developing a heart disease over time. If you're trying to drug it, it's not a good therapeutic target, as the pharmacology world and as pharmaceuticals will tell you over the last decade, but it still provides a tremendous amount of diagnostic information. So it's about really understanding the objectives, understanding that we're all, again, while we're all equal, we're not identical, and trying to understand how our disease processes may be different in a way that allows us to specifically target our disease process. Now, I think, again, in the oncology space, biomarkers has proven to be transformative. And the example I always use is when I was in medical school, the way we diagnosed lung cancer or classified lung cancer was based upon its pathology. It was either non-small cell lung cancer, squamous cell. There was essentially three buckets of what lung cancer looked like. And that was based upon what the diagnosis was on a pathologic examination. When you took a piece of that tumor out, you put it on a slide, you look at it under a microscope, I can classify it as one of these three groups. And then over time, we realized that there's specific mutations, genetic mutations that occur in various lung cancers, EGFR being the first one that was identified. And then subsequently now, when we look at lung cancer, the way we classify lung cancer now, it's one of 40 different diseases based upon the specific mutations. Now, based upon those specific mutations, oncologists today will decide what specific therapy to give an individual. And so lung cancer went from a disease of three individual components to one now that's several dozen different components. And that component classification is exactly what dictates what drug you receive. And this is why the efficacy has gone up quite a bit for treatment of lung cancer.
Wow, it's an amazing story too of a positive outcome for sure.
The question is now, how do we extend this though, right? Because this worked real cancer. How do we think about this for other non-oncologic diseases?
Definitely. And we're, of course, always as a founder, where do you get the most bang for your buck? But certainly being able to see the applications of that technology across other spaces outside of cancer. So, here we're talking a bit about your passion for it, but let's talk about Sapient for a minute. So it's a spin out from the lab there at UCSD, literally from lab to launch, which we love, of course. But tell us a little bit about Sapient and how you guys are trying to bring that transformative technology to a different therapeutic space.
Sure, happy to. And then happy to walk you through this evolution. Sapient was founded around this idea that if we can better classify disease using what we call biomarkers in the same way we use genetic biomarkers to classify lung cancer, we can better align a specific individual with their specific disease process and ultimately understand the specific therapy that's best suited. And again, this is not a theoretical idea. Certainly, there's a tremendous amount of evidence in the clinical literature that when drugs are developed together with a biomarker, whether it be the oncology space or in other therapeutic areas, the approval rates go through the roof on an order of magnitude. This has certainly been borne out. We were quite interested in the idea that when we observed what happened in the oncology space and how genetics had transformed oncologic understanding and treatment, it was really around not classifying the host, meaning you or I, but rather our disease processes. As I mentioned, being able to understand how a tumor may be different from another person's tumor by the specific mutations that are located there. And of course, this works well for cancer simply because cancer is read out by genetic sequencing, where you can tell what the molecular drivers are and then the specific mutations. And the question is, what about those other diseases, heart disease, lung disease, and neurodegenerative disorders, liver and GI illness, all those other diseases for which genetics has not proven to provide the same type of information? How do we begin to classify those diseases and better understand them, meaning everything outside of the world of cancer, and even in certain cases in cancer? And we became very interested in this technology referred to as mass spectrometry. These are pretty big devices, and obviously, given your chemistry background, you're quite familiar.
Yep.
These are really amazing devices, bioanalytical devices that allow us to take complex biospecimens that are composed of thousands of molecules and decompose them and measure the actual abundance of each of these molecules that are present in a biological. So the challenge with mass spectrometry was the same one that was posed to sequencing about 20 years ago, and that it's an incredible technology, incredibly robust, very accurate and precise in its measurements. It's just too dang slow to do on a population scale.
Right.
And so when we launched our laboratory at the University of California, one of our real objectives was to take a mass spectrometer and simply make it go 100 to 500 times faster than it had ever gone before. And that's what the objective was. And we spent many years tinkering and prototyping and developing new hardware systems, developing new software systems. And as we were going through that process, we were slowly solving each of these bottlenecks in a way that we were able to continue to accelerate the process as a whole. And as we were doing this, there was a number of organizations that started coming to us, government organizations, academics, large foundations, the Bill and Melinda Gates Foundation, large biopharma organizations, where they started asking, can you help work with us in order to be able to analyze this large population of biological sample that we have from this clinical trial or from this epidemiologic study? And we began doing this work. And as we started doing this, we realized that there was tremendous amount of information, both diagnostic information, prognostic information, as well as drug response information that's encoded in these small molecule biomarkers that are floating around in our blood that could be detected by mass spectrometry. And again, this is not magic. When you go to the doctor, anyone who's gone to a physician for your annual checkup, they draw those 2 tubes of blood, the purple top tubes, and we typically measure about 15 things in those blood samples. And there's tens of thousands of things floating around in your blood. Why are we only measuring 15 of them? Essentially, what we were doing here is using these mass spectrometry systems to measure 15,000 things at once in that biological specimen. The simple answer is that as we measured more things, we were able to learn more things. We could tell who was going to develop what diseases over time, how people were going to respond to particular therapeutics, who is going to have a more indolent response to a disease process versus a more precipitous response to a disease process. And as we began to do more and more of this work, it was clear that there was a larger sort of opportunity here to bring high throughput mass spectrometry to drug development in a way that would provide services and aid in drug development and discovery across the world for many different organizations, whether they be, again, academic, foundations, governments, biopharma partners, et cetera. What gave rise to Sapient. And so Sapient was spun out with that exact idea.
Yeah. And my next question, and I'm just sitting here spinning on this, right, was to talk about technology and its application evolving over the next several years. But as I'm sitting here thinking, as a general public sort of person, I have access to gene testing and those kinds of things, right? Like I have a family history of breast cancer. So I do that testing every couple of years because it keeps evolving. But I'm like, the general public, can I just send you a vial of my blood? What sort of plans do we have? I know targeting this to drug development is a place to start, but what about the benefits to the greater population as a whole? Do you see it evolving that way?
Absolutely, Kelly. Fundamentally, what we've built is a tool, and this tool allows us to accumulate a massive amount of underlying data that then allows us to develop new diagnostics. So there's many phases to Sapient. First is, as you suggested, just being able to service those that are developing drugs. And much of our attention in our early years here has been on supporting biopharma organizations with a discovery as a service type of model here, whereby we provide services to them to analyze their biological specimens, whether they come from preclinical studies or clinical studies, help them make discoveries and return that information to them in a way that allows them to accelerate their drug development. At the same time, through our internal R&D efforts, as you can imagine, we're amassing a tremendous amount of data. And we have one of the largest human biological data assets in the world at this point, where we've analyzed hundreds of thousands of samples using these mass spectrometry systems now at Sapient. And that's allowed us to develop new diagnostic tests. And it's our hope over the next year or two here that we'll begin commercializing some of these tests and making them available to the public.
That would be exciting. To pivot a little bit, Sapient's team spans so many disciplines. You've got chemists, engineers, epidemiologists, physicians, to name a few. But as the CEO, how are you intentionally shaping the culture and efficiency of such a high-performing team?
Yeah, it's an interesting question. And there's many models by which you can build an organization and build teams. And my essential model has always been you go out and find the absolute smartest, most talented people that are really excited to solve really hard problems together as a team. And you put them in a room together, you give them really hard problems, and you give them a lot of food, and you just get out of their way. And that's really been the model that's always worked for me, whether it be on the academic side or in building Sapient. And so I have to say we've been incredibly fortunate to find just some world-class talents across each of these areas. And I essentially view my role as being the glue or the grease. And essentially, when I need to bring people together, it's my job to be able to bring those folks together and bridge syntax divides and communication. And sometimes I have to sort of grease wheels to make things turn a little bit faster. But honestly, the most part, it's a train that I'm just holding on to. And it's my job just to help directly and make some minor tweaks. But we're very fortunate to have just world-class people that do all the hard lifting.
That's incredible. I love that story, especially about the food. As a
Yeah,
manager of people, I find that to
food
be
is
the
critical.
universal motivator.
Food and caffeine, a critical
Uh,
component.
yes, definitely caffeine as well. Absolutely. To talk a little bit about funding, you
Yeah.
mentioned you've raised funding from several sources, including the Bill and Melinda Gates Foundation. What advice do you have for other founders with funding on the mind?
Yeah, it's a really interesting question, Kelly. And we took a perhaps non-traditional approach to funding in that myself and my two co-founders initially, when we had launched Sapient, because we were a discovery and a service organization, we didn't necessarily need funding up front. We had clients who were coming to us, like you said, the Bill and Melinda Gates Foundation, which is public information. We had many other biopharma organizations that were working with us. And so we had revenue almost from day one. And for that reason, we weren't in a position that we required funding. Now, at the same time, we realized quite quickly that the demand out there for our services were far greater than we were going to be able to provide, given a small closet model. And we ultimately ended up finding a funding partner that really worked for us. And the approach we took is that we were going to find a partner more than anything else. And in funding, to me, the dollars that come in are only one component of it. Essentially, you're marrying your early investors, and you have to be okay with that, good, bad, or ugly. And we spent a lot of time making sure that we found individuals that were aligned with our larger vision, that complemented many of the weaknesses and areas that we did not have strengths in, and that were excited to go along that adventure from beginning. And I realize that the title of this is Lab to Launch, and it's very different having technology and having a successful organization or company. Those are two very different worlds. It's almost night and day, and those Venn diagrams don't cross very much. And optimizing technology, optimizing discovery, and optimizing companies and processes, two very different areas of optimization. And we are very fortunate that we were able to find an investor team that was excited about going through that process with us and walking us along from our lab as to how we actually build a successful company.
Yeah, I've spent the last, I don't know, almost 10 years working with startups in the space, and I've seen this go well. I've seen it not. So I love your humility in that because a lot of times what I see is the tech side person is the CEO and they just want to hold on to this and they want to control all the things. But at some point you scale to a place where you have to let go. You have to have a good team and you have to trust them to do their jobs and get out of their way. And so I love hearing how well that's going for you guys because I've seen it go well.
I could not agree more. In the end, I'm just one cog in a very large wheel, and the other cogs have to be able to function. Otherwise, the wheel doesn't turn. And the investor
Yeah.
team are part of that. Your board is part of that. Your heads of department are part of that. And everyone who works in our organization is critical.
On a bit of a more personal note, if you could go back to the start of your career, what would you tell yourself based on what you know now?
Oh my goodness. I think one of the key lessons I've learned in life is that you don't have to know what you want to do next. And in life, in my career, at least personally, has been really quite enough. And when I was practicing medicine, I was convinced that's the only thing I ever wanted to do. And when I was a professor and running a research lab and teaching, I was convinced that's the only thing I ever wanted to do. And I really enjoyed it. And now here on the commercialization side, running Sapient, I'm convinced this is the only thing I ever wanted to do. I think I've learned enough that I really have no idea what's going to happen next. And that's okay. That's part of the evolution of learning. And I think folks who are bright, who are talented, who are hardworking, there's lots of opportunities and there's lots of things you can do and not to be scared at the idea of transition.
Nice. I like
And
that.
that's the one thing I would say that I've learned over and over again, that it's going to be okay. And it's okay to try new things and it's okay to do new things. And you should never let fear be the driving emotion behind any idea or any ounce of effort.
I like that. Very true. There was another guest earlier, and so this is a standard one I'm using all
Sure.
the time now. I love it. But if I walked into Barnes and Noble, where would I find you? What section would you be in?
Oh boy, bookstores are my weakness. And I just love the smell of the bookstore when you walk in. And I don't know why. I don't know if it's the paper or the oil or the ink or what it is, but something about it is magical to me. And I suffer, as I think I mentioned early on, from intellectual ADD. And I tend to wander, and I wander extensively. And everything from just thinking about what's on my bookshelf right now or my side stand that I'm reading, everything from deep science and a book about the genome, to a book on regulatory affairs and drug development, to a book on management consulting and how you organize teams. And so tend to be pretty diverse in where I am. What I will admit though, and something again, another lesson that I've learned is that oftentimes we're quite disparaging of social media and learning from alternative sources. And there's nothing like holding a book in my hand. So I'm certainly a firm believer in that. At the same time, there's a tremendous amount of information to be learned from alternative sources, whether it be podcasts such as yours, whether it be Twitter and other areas. And I'm continually amazed just how much one can learn when you're exposed to such diverse ideas and opinions and thought leaders on these different types of social media platforms. And so I have to admit, a huge fan. I openly admit I've probably learned more in the last year on Twitter than just about anywhere else. And there's an incredible amount of information around gap accounting and how you think about P&L that come from really insightful people. And I find a lot of that information is just on Twitter. And so the simple matter is I'll be wandering around the bookstore, but I'll probably have my phone out at the same time.
Yeah, same. I found that LinkedIn has become a bit of a rabbit hole for me, which is not something I ever expected. Generally, I go to those sorts of things to escape my job.
Right.
I want to get out of my head for a little while, but man, same kind of thing as Twitter. And I don't spend as much time there, but LinkedIn too, I have found myself scrolling and then following threads and reading people's thoughts and opinions on things. And yeah, for all the drama around it, it certainly is also an interesting way to see a bigger perspective on the world than just your own local area or local people that you interact with.
That's exactly right. I couldn't agree more. And you can really connect with thought leaders and people that the problem with books, obviously, is that it takes years to go from inception and writing of a book to publication. And social media has taken that and accelerated sort of distribution of information and knowledge and opinions, for better or worse, is arguable, to an instantaneous time point. And that's really interesting to think about.
Definitely. Definitely. Where can folks go to connect with you and follow along with Sapient's progress?
Our website's probably the best place to go. And we have a marketing team here that does a great job of putting together white pages and blogs and publications and other types of information on our website. So www.sapient.bio. And there's a lot of information on there about what we do. And we're always looking to connect with people who are interested in these types of tools and technologies, whether they come from biopharma, foundations, disease organizations, government or academics, or even consumers that are interested in particular types of testing.
Awesome. Thank you so much for your time today, Mo. It's been a fun conversation.
Thank you so much, Kelly. Appreciate it.
Thank you for listening to this week's episode of From Lab to Launch, brought to you by Qualio. If you like what you've heard, please subscribe and give the show a positive review. It really helps us out. For more information about Qualio, our guest today, or to be a guest on a future episode, please refer to the show notes. Until next time.